Okay? Again, part of the cardiovascular system, right? Okay? Some of the functions, so we'll start off nice and easy, right? Some of the functions of blood. We know that it transports things. We've seen this before. It's a big transporting system, if you will, highway system that carries stuff to tissues and back to tissues and cells and back and forth, right? It's a regulatory type of system as well, right? Because blood also is a regulation system. And we'll talk about that also, right? And protection, it provides protection as well, right? And we'll talk about that as well. Like the white blood cells are part of the immune system. That provides protection for the entire body. So what do we mean by transport, right? Of course, nutrients are one big way we can transport through blood, through the composition of blood. Oxygen is a really important type of chemical that we use to make energy, right, as well. What about waste, right? Blood also carries waste from cells to places where we actually detoxify, break them down, and then eliminate them from the body. And mainly that's to the kidneys and the lung, right? Lungs take out waste. And what waste do we have that we're taking out into lungs? What's the main waste there? Does anybody remember this? What's the waste there that we're taking out? If oxygen gets used to make energy in cells, then what is the side product that we take out from that, making the energy that we take to lungs? Nobody? Carbon dioxide, right? We're using carbon dioxide as a waste product and we're taking that to the lungs. There's a reason why you breathe in oxygen and you breathe out carbon dioxide, right? And the kidneys eliminate also waste, okay? And one of the ways we do that through urine, right? If you ever notice the color of urine, right, yellow? Well, the color of urine being yellow is actually from the breakdown of metabolic types of molecules that the cells are breaking down. And in particular, protein metabolism. So when we break down proteins, the side product is an amine group, an amino acid group, right, that we can't reuse, and basically it resembles a yellowish color, and when we add it to water, that's what we see mainly in urine, right? That's why urine is yellow, in fact. But then you can change the water content, you can make it more diluted, so the color of yellow, if it's really dark, really bright, or whatever, can dictate a lot of other things. But that's what we're doing, right? And then, of course, hormones, right? We just saw that in the video there. We saw that very briefly. We saw the hormones getting flooded in the bloodstream. Regulation, right? Body temperature, right? We can absorb and transport heat through blood, right? And we saw this, for example, in the integumentary system in AMP1 when we actually see how the skin cuts off blood supply when we want to conserve energy when it's cold and our skin feels it's cold or senses cold. And then we can also, on the opposite end, open up capillaries to let blood pool up into the skin so that way the heat can distribute from blood to the outside of the body. So that's also something that happens in blood. Now, we can actually regulate our pH using a type of protein we call a buffer. Okay? So there are buffers in our bloodstream that balance out our pH. Now, what is a buffer? A buffer is a type of, and it's usually a protein, it's a type of protein that will If the blood is too acidic, that same one protein will make it more alkaline. And if it's too alkaline, that same buffer will make it more acidic. So it can go both ways. Does that make sense? It can go both ways. Does anybody remember the pH of our blood? We know this number or this range. .735 to 745. 7.35 to 7.45 is the pH range of blood. It is what we call slightly basic. Okay? Make sure you memorize that, like burn that in your brain, because those in healthcare will have to know that. That's the pH range of blood. 735, 745. Okay? Well, then our buffers in our blood balance that, keep that in that range, basically. Okay? And then to maintain that fluid volume in the circulatory system in blood, to maintain the amount of water that blood needs, right, is done by the blood system itself. It's not really a system, it's part of it. And what do we mean by protection, right? So we have a kind of system in place to, in the event that when we break a blood vessel that holds blood, we have our way to actually block that or hold that blood inside the vessel and prevent extra leaking out of the blood, and we call that forming a clot, right? So we have the capability of forming clots in blood when we need to, when there's a breakage in a blood vessel, in fact. That's part of protection, because we're protecting blood loss, right? And then also infection, right? We have a lot of different properties of the immune system that we find in blood. Okay? One of which, yesterday at least in our lab here in Uvalde, we took a look at blood typing, right? And we took a look at blood types. And in fact, what we added to the blood itself was what we call an antiserum or an antibody, right? So depending on your blood type, you will have antibodies in your blood to prevent, for example, other types of blood from contaminating your blood. But we also have antibodies for a lot of things. So we create antibodies for various different foreign things that come into our body. And what they do is they bind to the object or the surface or the living organism and cause it not to invade other tissues. We've got proteins that act as things that neutralize foreign chemicals, foreign agents, foreign things, including bacteria and viruses. And of course, we've got our white blood cells. White blood cells are specialized types of immune system cells that actually do specialized responses, and we'll see that in this PowerPoint as well. So this is, we're looking at the composition of blood, right? We're looking at how it looks, what it's made up of, okay? So blood is in fact not just, a bunch of cells, it's actually a tissue, right? We considered it a tissue, right? And if we go back to the tissue portion of this of A&P, right? We know that this type of tissue, blood, falls into connective tissue, right? It's a type of connective tissue. So again, there's epithelial, connective, muscular, and nervous, if you remember those from A&P 1, right? This falls under the connective, and it's the only fluid tissue out there that we have in our body, right? So if it's connective tissue, you know it's made up of cells and a matrix, right? Like every other connective tissue has. And its matrix is plasma, okay? Plasma. Their cells are what we call formed elements, right? And under that umbrella of formed elements, there's a lot of those cells, right? We know that cells are placed in plasma, right? They're suspended in plasma. We would call that a solution, right? And by definition, a mixture, it's a mixture called a solution. And by definition, if we stop moving blood around, eventually the solutes, which are cells, will fall out of the solution, right? So what do I mean by formed elements? We've got the erythrocytes there. Those are your red blood cells, or RBCs. Your leukocytes, white blood cells, or WBCs, and platelets. Those are the three major types of formed elements that you have in blood. So in talking about blood and looking at its composition, if we were to take blood, for example, right, and spin it, right, we use a centrifuge, right, but you would probably do this in a lab if you're a phlebotomist or you actually look at blood every day to analyze it for healthcare. And if you're to do this, right, spin it, use the property of centrifuging, and spin it for a while and then let it sit, you would see these three separate layers. That's why we see this, right? The erythrocytes are always going to be at the bottom. And in fact, 45% of the volume, give or take, of blood is going to be the erythrocytes, or the red blood cells. In fact, hematocrits, or the word hematocrit, or hemocrit, stands for basically measuring the percent of the blood volume that is the red blood cells. That 45% is the hematocrit volume. So in fact, you can measure this, right? You would send this to the lab, so those in allied health that are dealing with patients that will pool blood, for example, to analyze it, we'll send it to a lab, and the lab will do this, and then they'll say, okay, the hematocrit level is this percentage. And it should be around 45% normally speaking, but if it's plus or minus, if it's higher or lower, then that could tell you something as a clinician, right? I mean, that's data, that's information that tells you, okay, something, why is there a lot of red blood cells? Why is there too little? Well, there can be very different, reasons why that would be true. In fact, when you look at gender, males versus females, males tend to have higher, on the average, red blood cell or hematocrit level than females do. Okay? Now, there's a major reason why, right? And the major reason why is because males have more what? This is critical thinking, right? So you got to put it together with what you learned in A&P 1 for that answer. Why do males have more red blood cells, is what it's saying here, factually, than females do?
They have more testosterone.
They have more testosterone?
Yeah.
That's a very male thing to say. No, I'm just kidding. But that's, yeah, not necessarily true. I mean, that's true, but I mean, that doesn't cause what we're seeing here. Why do we have more red blood cells in males versus females? We study this in chapter, 6. Skeletal muscle. Males have more skeletal muscle than females do, pound for pound. What I mean by pound for pound is if you had a 6-foot man and a 6-foot woman, same weight, same height, same age, and you measure their skeletal muscle, males will always have larger skeletal muscle than females do. Because they have more muscle, they need more red blood cells to carry more nutrients and oxygen to more muscle. Because of which, males have more red blood cells than females do on the average. Does that make sense to everybody? That's the answer right there. Right? That's what I was looking for. Okay. Now, white blood cells and platelets would be found towards the middle of the tube here, in a place that we call the Buffy coat. Less than 1% of the volume of blood is from the Buffy coat. And towards the top, or the top portion, is the plasma. And that's 55% of our blood volume is the plasma itself. So here's what we mean. This is a centrifuge, right? So yesterday in our experimentation, we didn't get to do this. We didn't do hematocrit. We don't have all the equipment to do this, and blood has its own precautions, right? Okay. But if you were to draw blood like you would do here in a lab, you would spin it. That's a centrifuge right there. And then you'd spin it for a while, and then we'd stop the machine, pull the tube out, and this is what you would see. This yellowish color on the very top, in the middle this whitish color, in the middle red. What we've done is separated all the formed elements in plasma by doing this in the machine, and this is what we see. And then 55% of the top portion that's yellow is plasma. The middle portion is the Buffy coat. That's where you would find your white blood cells and your platelets. And at the bottom, you would see all the red blood cells, all of them kind of squished up together. Okay? So just looking at then the characteristics of blood itself. Blood is characteristically sticky. It is opaque, okay? Has a metallic taste to it. Now those are all things that you could pull out, the information you could pull out to understand what's in blood, okay? So first, the first characteristic, sticky. The stickiness comes from the platelets. Now the platelets are responsible for the clotting of blood. And that if they get onto a surface, like for example, your fingers or your skin level, blood starts to stick together. And that's what blood does because of the platelets, the composition of blood that having platelets inside of it, the platelets help stick stuff together. Now, that doesn't happen in blood. Blood is not sticky inside of the blood vessel. It kind of runs really smooth and it doesn't stick. Once it gets to another surface, like for example, when you cut yourself, you break a vessel, it starts to stick all the way around it and then against the sidewall. And that's what it does. It kind of forms a stickiness. The opaqueness, right, which means it's not too shiny, comes from the red blood cells. But the red blood cells don't have this kind of shiny appearance. It's more of like a very red, dark fluid. And what about the metallic taste? The metallic taste comes from the iron that exists in and on the red blood cell. And we'll take a look at how iron gets used here. Okay? Now it's true when they tell you you need to eat iron-rich bloods. Iron is important as part of your diet. I mean, you've heard this before, right? Iron. And that some foods contain iron, like meats contain iron, right? But other things like vegetables, like for example spinach, contains iron. That's why we invented a character called Popeye, right? Eats a spinach and gets what? Big, right? You've heard this before, right? Popeye eating spinach. And the reason why is because it contains iron. Iron is essentially important to bind oxygen to the red blood cell. We'll see this in just a second. So we'll kind of keep that in mind when we get to that point. So what about the oxygen content? There are occasions in the body where blood is going to have a lot of oxygen and blood won't have as much oxygen. And why is that the case? Because when we breathe in, for example, when we inspire in our lungs, when we take a breath in, what we are doing is loading the blood with oxygen. And in the lung level, oxygen is transferring from the lung tissue that we just took in, all the oxygen, into blood. And then blood and red blood cells, in fact, bind the oxygen to itself. And when it does that, this is where that part of the bloodstream has a lot of oxygen in it. Now let's look at the other side when there's low oxygen levels. When there's low oxygen levels, this comes from the blood dropping off oxygen to the tissue level. Okay? And the red blood cells don't have oxygen on them anymore. They will be low oxygen type of blood. When we look at that, that's usually high oxygen level blood is usually kind of the scarlet red, bright red color. And usually the low oxygen level is a dark red, and sometimes we call this blue or purple blood. Some people describe it that way. It's a darker red. And it's just because the oxygen is not present there as much as the red, bright red color. So you can definitely tell if somebody's bleeding, for example, what kind of blood vessel you broke, because some of the blood vessels have low oxygen blood, and some of them have rich oxygen blood. It's like your arteries and veins. Does that make sense? It's kind of what you see there. So there's the pH, right? There's that range that I told you to make sure you memorize, 735, 745. Blood makes up about 8% of our body weight. So take your body weight, multiply that by 0.08. That's how much pounds of blood you have in your body, right? That's how much blood you have. We can also measure it by volume, right? So males have 5 to 6 liters, females have 4 to 5. And again, that's also because of the reasoning, right? Between skeletal muscle, more demand for more red blood cells, more demand for blood, right? Okay, so what about the plasma, right? Plasma is the actual fluid of the blood, right? That's what plasma is, the fluid. It's very yellow, straw colored is what we talk about that being, it's not a bright yellow, it's not a mustard yellow, it's kind of like a hay color, right, of yellow. This is where the stickiness comes from, because in plasma is where we have the platelets, right? That I told you that makes things sticky. That participates in clotting. But look at the percentage here, right? About 90% of plasma is water, just pure 100%, H2O. Vast majority of it, 90% in fact. What about the rest of the 10%? About 100 different solutes in there. Nutrients are of course found in plasma. Gas is found in plasma, like the oxygen, carbon dioxide, and other things, other gases. The hormones that we talked about when we release hormones in the bloodstream, they're found in the plasma. The waste that we talk about is found in plasma. Proteins, ions, all those chemicals are there. So when we separate them, if we separate them, right, kind of going back to this, and we separate them, we can look at the plasma and test the plasma for hormone levels, right? Kind of going back to the endocrine system. And then we can test hormone levels there, and we can say, okay, this range is a good range for this type of hormone, but why would you have too much or too little? So a blood value or blood testing can tell us hormone levels, how many ions you have, how much oxygen you should have, how much carbon dioxide should be there, how much waste is there. And if those ranges are off, then we know something is not 100% working efficiently in the body. That's why we're looking at this. Okay? The plasma proteins are the most abundant solute that you have in plasma. So out of the 10%, the majority of what you have there is plasma proteins. Plasma proteins, okay? They stay in blood. They don't get taken up by the cells, so they don't go to the cell level. They stay in blood. They're huge, by the way, in size, right? Those proteins are made up mostly by the liver, so the liver makes the proteins and then puts them in blood, and that's how we have the proteins there all the time. Those proteins will have a shelf life, and then after they expire, then the liver will break those proteins down and then put in new ones. Okay? Now, out of all the plasma proteins that we have, the one that we have the most of is a protein called albumin. Okay, albumin. And albumin does a lot of interesting things for our body, right? It carries molecules in our blood. It is one of the buffers of blood. And it gives us blood pressure, helps us maintain blood pressure. Because adding proteins to fluid actually adjusts or influences pressure in a fluid like blood. Okay? But the one that's really important is it acts as a buffer. It actually balances the pH. Albumin. So this table here just shows you the different types of other solutes other than albumin. Albumin's there, but there's other proteins there, and they show you the proteins there. Then they show you some other solutes there, and then the water portion of it, right? There's that table there. Okay, so we'll discuss then the form elements, right? Okay, well, these are the actual cells, or what we call fragments of cells or pieces of cells. Okay, so we have two different types of whole cells, cells that are complete. The red blood cells and the white blood cells, or RBCs and WBCs, right? So when I mention cell pieces or cell fragments, what I'm mentioning is the platelets. Because in fact, platelets are pieces of a cell, right? And we'll see how platelets form as well. When taking a look at white blood cells, they're the only ones that have a complete cell. And what I mean by that is white blood cells are the only cell that has a nuclei. Since platelets are pieces of cells, they don't have a nucleus. And red blood cells do not have a nucleus. They're denucleated. On top of that, they don't have organelles as well, which is interesting, all right? And we'll see why in just a second, so we'll look at that also. Most formed elements only last a couple days in the bloodstream, right? And they have to be remade, reprocessed. Red blood cells last for a while, but they also have a very short lifespan. Most of your blood cells will originate in bone marrow, right? And they don't undergo mitosis, right? So in the bone marrow is where we have blood cells getting produced, right? Which we already know because we've taken a look at bone tissue and AMP wall. Here are your blood cells, right? So we see an electron microscope picture there, sorry. An electron microscope picture there on the very top. And you see all of your different formed elements, red blood cells, white blood cells, platelets. But you can also identify them at the bottom when you look at a blood smear. And in lab, yesterday in lab in Uvalde, we took a look at a blood smear, so we took a look at exactly what this looks like at the bottom and identified red blood cells, which are kind of a donut shape kind of cell. And then the larger ones that are kind of have this bluish purple color, larger cells, those are white blood cells. And if you notice, there's little tiny dots in between, and those are what we would call the platelets. Okay, so we'll talk about red blood cells. Talk about how they're put together, their function. Okay, and then at that point we'll move on to another topic, right? So their main function, erythrocytes, red blood cells, okay, is for gas transport, right? And in particular, their main gas that they transport is oxygen, right? Oxygen is what they transport. They also transport carbon dioxide, but not in big amounts, okay? Looking at shape, and then a red blood cell has a biconcave disc, which means it's like a bowl. It has a little bowl shape on one end and the other end, okay? That's what we mean by bi, means two, concave disc, which means the bowl shape is shaped on one end and the other. And we'll see a picture of that. It's also a picture of that in your book. The cell inside the cell, because this is actual cell, so it has a plasma membrane and all that. The cell inside is filled with mostly hemoglobin. Now, hemoglobin is a protein, okay? It is what we call a quaternary protein, which means it's a complete protein. And we'll see what a quaternary protein is in a second. And the hemoglobin is what actually binds to oxygen. Okay? This is what transports gas. Oxygen being mainly the one, but you also have carbon dioxide. And you can also load it with other gases. It's just those are the ones we kind of just focus in on. Okay? Now the diameter, the 0.75 nanometers, micrometers, the diameter is sometimes larger than a blood capillary. So what is a capillary? It's the smallest blood vessel we have in the body, right? That's what a capillary is, if you didn't know what a capillary was. Capillaries, those are the smallest blood vessels that actually drop off the nutrients and pick up waste to the cells, right? We have bigger blood vessels. We call those arteries and veins and all those other things, but the smallest ones we call capillaries. Red blood cells and their diameter are larger than the capillaries. And because of that, they can actually fold in, kind of like a taco, if you will. They can fold through those capillaries and squeeze right through. Their capability of doing this is because they have this protein. We call this spectrin in bold there. Spectrin is a protein that actually makes it really, really flexible, the cell flexible. It can fold and bend like it's doing there when going through the capillaries. So this is a really good example of how anatomy, the structure, and the function and physiology work together in a red blood cell. Because of its shape, because of how it's put together, it has a really good function, which is gas transport and moving and bending and what have you. The biconcave, the little bending or the bowl on one end and the other of a red blood cell, gives it a greater surface area and will allow for more oxygen to be binded to one red blood cell. Since hemoglobin is the protein that actually binds to oxygen and carbon dioxide, most of the volume of a blood cell is made up of hemoglobin. Now, a mature red blood cell will not have organelles as well, including mitochondria. And we know that mitochondria's responsibility is to produce what? Make ATP, right? So that means they don't produce energy for the cell. Okay? Which means they don't need the oxygen that they're carrying to other cells. They don't need it to make energy. That means that when red blood cells bind oxygen, it doesn't need energy, and it doesn't need energy for the red blood cell to travel to the cells. It just does it. It floats with pressure and then drops off oxygen. So it's very much just a vehicle for gas exchange. That's its main function. So it doesn't need organelles. It doesn't need a nucleus. It doesn't need anything. It just needs the shell. It just needs the cell, the shape and the plasma membrane. That's basically all it needs. And a bunch of hemoglobin. So here's a side view. This gives you a great example of what you can see. We call this a little biconcave disc here, because this is the red blood cell, and we're looking at a side cut, cross cut, to see the little indentation, little bowl on one end and the bowl on the other, right? Then we're looking at the top view to see the kind of little, looks like a donut, sometimes looks like an inner tube, if the inner tube was filled up in the middle. It's not an empty hole, right? That's what we see there. So again, gas transport is their main job. We talk about respiratory gas transport because that's where it's coming from, the respiratory system, right? We're taking in oxygen through the lungs. Red blood cells go right by the lung tissue, and then oxygen diffuses. If you remember diffusion from back in A&P 1, high concentration, low concentration, oxygen diffuses into the red blood cell, or to the bloodstream into the red blood cell, and binds immediately to the hemoglobin inside the red blood cell. Just like it binds, oxygen can also unbind. So it can bind to the protein and then it can unbind there too. So there's a value here, right, for males and females, right, of red blood cells, 13 to 18 grams per 100 milliliter of fluid, of blood, and 12 to 16 grams per 100 milliliter of blood. And those are values that are normal, right? And again, when you take blood and you run samples and then look at the results from a lab, which you will do as a clinician later on, many of you, then you can already tell, okay, these are the ranges, right? So you'll memorize these ranges eventually when you get most of you get in nursing school, you'll memorize them to heart. But then after a while, the blood values, the panels, they call them panels, but the reports come back and they give you the normal range. So it's good to memorize, but you'll always have a, somewhere to reference back to. Right? So what is hemoglobin? Again, it's a protein, but what is it consisting of? So there's a couple things it's consisting of. And it shows you that in bold there it says heme group, right? That's the pigment molecule. Okay, heme. And that pigment molecule is also bounded to the protein globulin, right? Or globin. When you add the pigment and the protein together, we call that hemoglobin, right? That's where that word comes from, hemoglobin, right? The pigment and the globin molecule. Now, the protein, globin or globin, has 4 polypeptide chains. Polypeptides is amino acids binded together. And poly means what? 3 or more, right? That would be a polypeptide. Many, so and it's a big amino acid. So this is way more than just 3. And there's 4 of those chains binded together. And when we talk about a quaternary structure, that's many tertiary structures, at least 2 or more, binded together. That's what we call a quaternary structure. That protein's a quaternary protein. So 2 of those chains are alpha chains and 2 of those chains are beta chains. We'll look at them. We don't need to concentrate so much on the beta and alpha thing, but just to show you that they're different polypeptides binded to make one hemoglobin. And in the center of each polypeptide, okay, is one hemipigment molecule. That molecule gives the characteristic red color to blood. Because in fact, what you are looking at in blood that makes it red is the hemi group on the hemoglobin inside a red blood cell of a red blood cell. That's what makes it its characteristic red color. And if you remember when we talked about the integumentary system in AMP1, we talked about what contributes to what? Skin color, right? Melanin, right? Keratin, but also what? Hemoglobin, right? That's what we're talking about. The pinkness on your skin level, right? How, you look like this, you got this color, but also the pinkness, right? The pinkness is the hemoglobin in the red blood cell. Now, the function of a hemi group is not because of the color. It just happened to be that color, right? It's really because that hemi group is the one that binds to iron, and then iron binds to oxygen. That's where we held iron on. So I want to go to the figure here because the figure really shows you what this is, right? If you notice that there's the bottom there, you see this 10, 10, 2, 10 little kind of squiggly lines that kind of go back and forth and kind of, are on the bottom here. And these are called the alpha groups, right? There's 2 of those. Those are the polypeptides, by the way. 1 and 2 at the bottom. You can see them kind of separate, but they're binded together. And the top darker brown color is the beta chains, beta globin. And there's 2 there, right? But they're all binded together. All 4 of them are binded together in a chemical bond. Okay? In the center of each one of those, you can see the red circle with the red lines. That is the hemipigment binded inside each of the 4 polypeptides. And in the middle, you can see this kind of green circle, and that's one iron molecule. And what they've done is on the right side, part B, they've blown it up, and you can see the chemical structure of a hemipigment. And in the middle, it's binded by an iron group. One oxygen molecule, an O2 molecule, binds to iron right in the middle. Okay? So if there's 4 of them, that means one hemoglobin can bind to 4 oxygen molecules. Does that make sense? So do a little math here, just a little quick short math, right? Again, one hemoglobin molecule binds to 4 oxygen molecules. We actually, in fact, for every 1 red blood cell, we have 250 million hemoglobin molecules per 1 red blood cell. So if 4 can bind to 1 hemoglobin molecule, how many molecules of oxygen can be binded by 1 red blood cell? How many? Not 1,000.
A billion, I'm sorry.
A billion, yeah. You didn't carry too many zeros. A billion, yes, very good, right. A billion. 4 times 250 million is 1 billion. 1 billion oxygen O2 molecules can be binded to 1 molecule of 1 red blood cell. that make sense? So how many red blood cells do you have on the average? About 6 million per 1 milliliter of blood, or 6 million per 1 liter of blood. So there's a lot. That's what I'm saying. A lot of oxygen gets used, is what I'm saying, for all the cells of your body, because oxygen is used for the creation of what? ATP. Does that make sense? This goes to show you how much oxygen there is, or can be carried. Okay? When we put oxygen onto a red blood cell, okay, we call that an oxyhemoglobin, which is just a fancy word to say that the hemoglobin has an oxygen binded to it. And oxygenated blood, talking about oxygenated blood, is this ruby red, scarlet red color that you notice, right? That's oxygenated blood. That's blood that has oxygen loaded onto it. When we unload it, which means the red blood cells have to go to the tissue level and then give off oxygen, unbind it, and then send it to the tissues in the cells, then we talk about being that deoxyhemoglobin, where the hemoglobin does not have oxygen on it. And it usually is a signature red color. I'm sorry, a dark red color. Or blue blood, or purple blood, or sometimes some people describe it as black blood. It's so dark. There's two distinct kind of things. Now, oxygen can also bind carbon dioxide to it, but only 20% of the carbon dioxide gets binded of the carbon dioxide that we make in our body. Only 20% of it gets binded to hemoglobin, or the red blood cell. Only 20%. The rest of the 80% is found in the plasma of blood, and the buffers break that carbon dioxide down. So the process of hematopoiesis is the process of making all the blood cells of our blood. That's the process of hematopoiesis. This occurs, and we've got to go back to what? Bone tissue and bones in the red bone marrow. The red bone marrow is responsible for making all of the blood cells that we have in blood. And again, just to go back to skeletal muscle, or skeletal system, the axial skeleton, the long bones, the girdles, right? And the humerus and the femur, the proximal epiphysis, where you find spongy bone, if you remember that. That's where red blood cells are getting produced. Or I'm sorry, all blood cells, not only just red blood cells, but all of them. In that area, in bone marrow, you have these stem cells, right? Hematopoietic stem cells. Or hemocytoblasts, that's what we call them. They are stem cells that can divide and turn into all formed elements. Completely fantastic, amazing how this works. These one sets of cells can turn into anything, any type of blood cell we need. If we need more red blood cells, they'll turn into red blood cells. If we need more white blood cells, they'll turn into white blood cells. If we need more platelets, they'll turn into the cell that we break apart that turns into platelets. That's hematopoietic stem cells. They're dictated to turn into any blood cell that we need, that we demand at this point, at any given point in our body. So who dictates how, when, and how these form, or which ones are which? Well, hormones do that, right? Growth factors as well, but hormones dictate growth factors and what have you. Once the stem cells decide that they're gonna turn into whatever cell they're gonna turn into, they cannot undo that. They begin a one-step process. That's a one-way system. After they became whatever they become, then they go into what we call blood sinusoids, which are little collection areas in bone tissue, and then they get shipped out into the bloodstream eventually. Let me go back really quick. The basophilic erythroblasts at the very end are making a bunch of ribosomes. Once they have made a bunch of ribosomes, they're called basically a polychromatic erythroblast. And these do what? Make a bunch of what? Hemoglobin. That makes sense? That means all the ribosomes are set up and the ribosomes are just pumping out hemoglobin, 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 hemoglobin. Once they've made hemoglobin, 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 in the appropriate amount, then the next stage, this orthochromatic erythroblasts, have just pretty much just hemoglobin inside of the cell. So what is going to disappear? What's going to fall away? Well, most of your organelles that are going to go away, the nucleus degrades or kind of shrinks up and gets eaten up by the cell. And it starts to pull in and becomes the concave shape, that biconcave disc. After this short process of this happening, we call the next step the reticulocytes, which still have a little bit of ribosomes. And at this stage is about 2 days of the 15-day process, this 2-week thing. Because it takes all 2 weeks to have 15 days for this to occur on the average. And after that reticulocyte, it becomes a mature erythrocyte. The ribosomes that are left over completely degrade, and it's pretty much a shell of a cell, plasma membrane, with a ton of hemoglobin inside of it. Now, why do I want to talk about this and why do we like discussing stages of red blood cell development? Is because sometimes you have diseases that will stop the process. That makes sense? And we can see that evident if we pull blood or we look at tissue, we can see the blood and stopped in these stages or they misform. And then this is the type of situations that you would see, for example, like anemia. When we saw anemia yesterday in the slides, the anemia in lab, then that's dictated because there's probably issues going on with these stages of making red blood cells. So this is what you see. Here's that figure 17.5, that shows you the process of hematopoietic stem cell, proerythroblast, basophilic erythroblast, polychromatic, orthochromatic, reticulocyte, and erythrocyte. I just want you to look at orthochromatic erythroblasts. Sorry, orthochromatic erythroblasts, right? You see how it's excising or shipping something out of its cell? The little purple dot, what do you think the purple dot represents?
The nucleus.
The nucleus, very good. Yeah. Good. The nucleus is getting shipped out. You see what I'm saying? It's breaking it down and then shipping all of those organelles and pieces plus the nucleus out of itself. It's making room basically for all the hemoglobin and everything gets shipped out, and this is kind of the middle step, the reticulocyte, and then we become a erythrocyte. This is what we're seeing here. So what happens when you have too few red blood cells, which can't happen, right? We call this process tissue hypoxia. Hypoxia is less oxygen or low oxygen, right? So in fact, we can talk about why that's the case. Could be because the person's not breathing correctly, so they're not transferring oxygen into the red blood cell. But we can also talk about the production. This is actually what we're talking about, right? If we have too little red blood cells that are mature getting produced, well, then you're going to talk about there's going to be fewer red blood cells that can take up oxygen and send it to tissue. And tissue basically is starving for oxygen. When they don't get oxygen, guess what happens to ATP? What goes down or stops? And keep in mind that cells in our body do not have a lot of ATP stored away because it doesn't work that way. We don't store it away a lot. We have a little bit left in our cells, but we consistently need to make ATP all the time, which means we consistently, us as humans, need oxygen all the time. Does that make sense? If we didn't need oxygen all the time, we could stop breathing, right? Just stop breathing for an hour or 30 minutes or whatever the case may be. And everything would be fine. We'd still carry on ATP production in our cells and everything would be great. But in fact, we need to breathe all the time, right? I mean, even sleeping, we slow it down, but we're still breathing nonetheless. Right? Does that make sense? I mean, we can only last a very short amount of time without oxygen. Right? I mean, we're in the summer and people are out in the water. Go try to dive next time and hold your breath under the water. See how long you can last, right? My record was 4 minutes, but that was when I was younger. But yeah, it's usually a minute, a little bit more than a minute, unless you're really physically healthy. You can last longer than that, but that's as far as we can do it, right? Too many red blood cell production can lead to viscosity buildup, right? Red blood cells can be thick, can cause the blood to be really thick, like sludge, basically, like oil, right? When you do that, that's what can occur. So here's the rate of red blood cell production. About 2 million are made per second. So we're not talking about just a small amount. We're cranking it up, basically. We really do a really good job of making a lot of it really fast. So it feels like it was a very slow process when we just looked at the steps, but this happens really quickly. Okay? Now, what influences this rate of production? Here we go with hormones, right? Hormones can dictate, right, when this happens, so we can control it by the hormones getting secreted. So there's hormones that actually are involved in telling the bone tissue to make more red blood cells or less or whatever. And the diet, right? The diet of the human is also a big influencing factor in red blood cell production. Okay? So talking about hormones, which hormone is one of the main ones responsible for the production or the stimulation of production of a red blood cell? Erythropoietin, or EPO for short. So there's a little bit that's left in our bloodstream. It's always there in a very small amount. Okay? When we have less red blood cells or less oxygen that is read by the kidneys, then the kidneys are the ones that will release erythropoietin. So the kidneys are responsible for releasing this, making and releasing this hormone. So what is in the kidneys that is doing the reading here, right? So there are enzymes that, are sensitive to oxygen, and when they don't get enough oxygen through the bloodstream, they do not break down this factor, this chemical. This chemical builds up in the kidney, and then shortly the kidneys release that hormone. Blood's in the bloodstream, goes to the bones, bone tissue, and then it stimulates those stem cells to divide. That's what occurs. Just a way for us to regulate production. So this just shows you how sometimes we have hypoxia forming in the body, right? Well, of course, if you're bleeding out, if you're bleeding, even bleeding out, which means a tremendous amount of acute bleeding, right? Or if you're slightly bleeding, because sometimes that can occur depending on disease and disorder. If hemorrhaging is occurring, then that could stimulate then red blood cell production. Or if you're destroying red blood cells, because sometimes that can happen, you can also cause this to occur, right? Also, if you have a very low number of hemoglobin molecules getting produced and put into red blood cells, this can also lead to hypoxia. And then, of course, the reduced availability of oxygen, which means if you have lung problems, if you can't get oxygen into blood, then that can also lead to hypoxia and can stimulate the body to produce more red blood cells. I want to take a look at the second bullet point really quick. Right? Insufficient hemoglobin per red blood cell. So I want you to give me examples of where you would see this. Right? The second one there. What situations mainly would you see an insufficient amount of hemoglobin in a red blood cell? Which means the red blood cell's formed, it's ready. That's not the issue. The issue is that the red blood cell doesn't have enough hemoglobin inside of it. What would be some reasons why you would have less hemoglobin in a red blood cell? Why would that be true? Where would you see that situation? And I'm going to go back just a second to give you a hint, right?
No, mom was anemic. That's the only thing I know is that she would just bleed out and it wouldn't even stop, especially like the clotting and stuff like that as well.
In anemia?
Yeah. I believe that's what she had. I don't know.
Maybe hemophilia. That could also be it. But here's the clue, the two bolded things down below. Right? So what, for example, could cause us to have an insufficiency of hemoglobin? Not red blood cells, but hemoglobin. The two factors that play a role in red blood cell production and hemoglobin and everything is hormonal control and dietary requirements. So what would happen, for example, in somebody that would be, I don't know, anorexic? Anorexia is, that's actually increasing in the United States, by the way, anorexia. Everybody knows what anorexia is, right? Okay. Psychological condition, but it also influences the body, right? Because the person doesn't eat enough, or in another condition, they will eat and then throw everything back up, which means they don't have enough nutrient absorption. How would that affect red blood cell and hemoglobin? Think about it. In somebody in, I don't know, anorexia.
The only thing I can think of is they don't have enough sufficient intake to create, mitochondria or ATP.
You're on the right track, but insufficient intake of what? What? Of what? Iron. Okay, that's one of them. So where would you find iron from? Food like what? Red meats and spinach, like dark green leafy vegetables. So in anorexia, if they're not getting the food in, if they're not refusing to eat, or they eat very small amounts, or they're eating and then throwing up, they're not getting enough iron in there. If they don't get enough iron in there, then you're not going to load oxygen on the red blood cell. You don't load oxygen in the red blood cell, you've got hypoxia. Very good. Number one. It's a great example. What else? In our anorexia patient. What else is important for red blood cell production or seeing the hemoglobins? What is hemoglobin made up of? Protein. So where in the world do you think we need that protein from? Where do we get it from? The food that we eat, yes? So in anorexia, if you're not eating protein or eating enough of it, guess what's going to be affected? Hemoglobin production. Because we use those proteins to break them down, build those proteins back up in the form of hemoglobin, right? And we got hemoglobin. And a person with anorexia, they're not eating enough proteins either. Iron is affected, protein is affected. Those are issues as well. Does that make sense to you? That's where diet comes into play, or poor diets, because it doesn't have to be anorexia. It can also be miseating the wrong foods. That's where balanced diets come in, and that's where each person has to have that balanced diet. That's why they talk. Does that make sense? You see what I'm saying? That's clinical kind of case study kind of thing, because when you look at those type of situations in a patient, nursing is about discovering the disorder and fixing the disorder, disease, whatever that might be, the imbalance. To help you understand the imbalance and how to fix it, you need to have a background on how the body functions normally. Yeah? That's what this course and the EMP1 is all about. How does the body work normally? What you're going to study in nursing for most of y'all is what happens when it goes awry, when there's an imbalance. Because your patient's not going to come healthy all the time, right? Most of your patients will come in sick with something. Does that make sense? The big mystery in nursing is to figure out, you're a nurse, but you're also kind of like a investigator. You're looking at everything and putting everything together and like saying, okay, it should normally work this way, but because I see something different, what is the abnormality there? What's the imbalance? The imbalance is this. In nursing, I know how to fix it. You prescribe this, that, and the other, this treatment, that, and then the person is better. Does that make sense? So that's why we're talking about these kind of examples. Okay, so again, EPO, erythropoietin, right, is the hormone that is responsible for initiating the production of red blood cells in that whole process. And again, on the opposite end, too much erythrocytes, too many red blood cells, too much oxygen in the body will then slow down the release of EPO. We can slow it down too. Now we talked to, somebody said hormones, right? But testosterone actually enhances EPO production. Okay? So not only is testosterone getting produced a lot in males, but there is a small amount also produced in females, and that's also one of the reasons why testosterone gets produced in females. Again, this is homeostatic control. That's really what we're talking about, right? Negative feedback system, homeostatic control, and this is what we're seeing. Our stimulus is hypoxia, low oxygen levels in blood, and what's the body responding with, right? Here's our whole kidneys, there's the bone tissue. We're looking at spongy bone. There's the proximal epiphysis of let's say that's the femur. It looks like a femur. And that's where that red blood cell production is happening. More red blood cells, more ability to carry oxygen, balancing the imbalances, right? So sometimes this happens. Athletes can use EPO. There's a synthetic version of it. This has happened textbook before in cases. I talked about Lance Armstrong. Lance Armstrong was also using EPO. Hate to pick on him, but he was using EPO as well. Okay. So EPO increases, again, stimulates red blood cell production, but it also increases hematocrit. And what is hematocrit? Blood volume measurement of red blood cells. And remember, that's about 45%, so it goes much higher. But what can happen if you take artificial EPO or stimulate that? Well, there's all the way up to 65%, and that can actually cause dehydration because it's concentrating the blood even more. There's no room for water, right? So we have dehydration. It can slow down blood flow, which we talk about sludge, but that's increasing viscosity. And that can cause clotting. It can cause blood vessels to break open like in a stroke. Or it can cause the heart to fail because the heart is working so fast or so hard to push out the slow kind of sludge moving blood. Those are the side effects of, again, taking artificial EPO. So here's the nutrition back again, right? You see nutrition there, right? Here are the requirements for the production of red blood cells, right? Here are your amino acids, so that's found in proteins, right? Amino acids are found, those are the monomer of proteins. But we also need lipids. Why do you think we need lipids for red blood cell production? What do lipids get used for? This is a real critical thinking one. All right. What are lipids get used for? Lipids get used for the production of plasma membrane. And remember, a red blood cell is all plasma membrane, right? The cell. So we need more plasma membrane. And other things, but mainly plasma membrane. And carbohydrates, right? Carbohydrates get used to produce ATP, right? Mainly. That's what we're using all those three things for. And guess what we have in a balanced diet? All three of those. Plus nucleic acids, but all three of those. We get iron from the diet, right? That's the only way we can actually take up iron. We don't make iron. It's an element. It's an atom. So we got to find it from other sources, and that's usually food, right? Take iron in with food. So 65% of the iron that we have in our body is in the hemoglobin molecules in red blood cells. The other amounts, we actually store it. And where do we store it? The liver, the spleen, and the bone marrow. In fact, if you're a fan of, I don't know, liver and onions, which I am not at all, but some people are, usually your organ meats are going to be kind of this minerally taste, metal taste, liver especially, if you eat liver, because that has a lot of iron in it. So, in fact, iron is not free in the bloodstream. It can actually be toxic. Iron is actually poisonous to the body. Okay? So what we do is we actually bind it to protein. So we have a protein carrier that carries iron in a cell, or it actually stores it in a cell and carries it when we need to carry it to, let's say, bone marrow, or to put it into a red blood cell, right? And these two, the ferritin and the hemostatin, are chemicals that we bind iron to in a cell. And then we call this other protein that we carry iron in blood transferrin. So talking about vitamins, vitamin B12 is a really important vitamin used for red blood cell production. So is another type of vitamin we call folic acid. Now, what are these two used for? These are used for cofactors and coenzymes for the production of DNA. Now, why is this important for a red blood cell? Because at a certain point of making red blood cells, they have to undergo what? Mitosis, right? And in the process of mitosis, you make copies, complete copies of DNA, right? Thereby, you need these two to make a lot of DNA in a red blood cell. So if you're making a lot of red blood cells, you definitely need these for the production of DNA, which is going to need to be into a red blood cell at the beginning stages. So the lifespan for a red blood cell is about 3 months or so. 3 to 4 months. And in fact, those red blood cells do not have a nucleus, so they do not undergo mitosis, right? So they cannot make proteins or anything. They can't grow or divide, right? So the red blood cell becomes fragile after a while. The hemoglobin begins to break down. That's right at the 100 to 120 day span. And usually at that point, they get trapped into the spleen. And the spleen is actually part of the lymphatic system. And in the spleen, we've got macrophages. Macrophages are big cells that actually undergo phagocytosis, right? Eating. And what they do there when they get trapped is that the macrophages will actually engulf, eat up the red blood cells and break them down. Because they can sense, they already know the ones that are actually becoming fragile and worn out and breaking down. So our body breaks them down through the macrophages in the spleen. So the old ones get broken.
Once they start breaking down, they're no longer read as an actual like blood cell body normal.
Normal, no, yeah, they're not.
So that's why they destroy.
Yeah, they get trapped in there, and then they get trapped. Macrophages break them, and then they no longer exist as a red blood cell. Right. So what do we do when we break them down? When the macrophages break them down, they separate the hemi group, the iron, and the globin or globin protein. The hemipigment, the iron, and the protein all get separated. The cell that's the plasma membrane, that gets all eaten up and gets broken down and it's all taken out. But those three parts are separated. Now iron gets bounded to ferritin or hemoseridin in a cell, in cells, and they get stored. So we reuse the iron. We don't need to get rid of it, we reuse it. It's really cool that we do that. And then we can take that iron and then put it back into new red blood cells. Now the hemi group, the pigment, does this really interesting thing in our body. The hemi is broken down by enzymes into another pigment we call bilirubin. Okay, bilirubin. Now who does this? The liver does it. The liver breaks down hemi into bilirubin. Bilirubin in the liver is getting secreted into the intestine. And we call that secretion of bilirubin into the intestine bile. Bile, right? Through a bile duct, okay? It's yellow at the time, and in fact, if you've seen bile, which means you've seen somebody throw up yellow kind of substance, that's usually bile. It's from the old breakdown of hemi, the pigment, in red blood cells. Now the intestines, the small intestine in fact, and the large intestine, break it down because they have enzymes that break it down to another pigment called urobilinogen. And then urobilinogen goes through another process of converting it into sterolcoblin. And that color is brown. There go. Feces is brown. Why is feces usually brown? It's because of the old pigment that gets broken down in all these steps into a brown pigment we call sterolcoblin. And it gets eliminated out of our body. So we don't reuse the pigment, we actually break it down in this process, which I think is pretty neat. This is what makes feces or poop or shit brown, right? This is what makes it brown. The globin molecule, the protein, is actually broken down into amino acids. Those amino acids can be released in circulation, and then our other cells can reuse the amino acids. Because those are monomers, right? That's the monomer of a protein. We can break that down. Which I think that's an interesting kind of thing where we break down the pigment into that. Right? So that leads me into another kind of thing that some of you will have experience in, is actually looking at poop in your patients. That is why they take fecal samples. That is why sometimes they do fecal recalls, which means the patient's in the hospital and you're the hospital nurse, and then you go collect poop samples. And maybe it's because you're actually looking at what kind of composition is the poop, but also the color. That's a big indicator as well. Which nursing can be sometimes a nasty endeavor, right? You have to be peed on, pooped on, you got to collect poop. That's part of the nursing, part of allied health, right? So this is the process, by the way. This shows you the process of what I just talked about. That whole process of recycling a red blood cell out of its 100 to 120 days. And this is what's occurring, involving all of these things right there. Okay, really quick, let's talk about these disorders, right? And one of them that we looked at yesterday in the slides is anemia, right? Krista said you should have looked at those. I think they have the, Mr. Pettis had those anemia slides there. So, and I wanted you to look at these because you want to look at how the cells look a little bit different, or a lot different, when you look at a normal red blood cell panel or smear versus not. So let's take a look at what anemia is, right? Anemia is because there is a low oxygen carrying capacity, right? Oxygen is not being carried to the cells and tissues, and it's already starting to affect the metabolism of cell. A cell cannot do what it's doing because it doesn't have oxygen. So it's more of an issue than a disease, right? We don't really call anemia a disease. It's more of a situation, right? It's more of a problem. And there's different types of anemias, obviously, right? We'll look at those really quick. But what is some of the symptoms that you would see if you were a clinician treating a patient that would have anemia? Well, fatigue, pallor means kind of just like very slouched over, slouched. Okay. Tiredness, dyspnea, chills, right? And because there might be these things involved, right? Blood loss can lead to anemia. Not enough red blood cells being produced, or too many being destroyed. So the in, the out, and then what about the whole blood loss, right? Could also be an issue too. So we classify anemias based on those three things, and these two are classified under blood loss, right? Hemorrhagic anemia talks about the rapid blood loss, right? This is somebody bleeding out, car accident, gunshot wound, stab wound, outdoor exercise, accident, many different things, right? Blood loss, right? Hemorrhagic anemia. The chronic one is going to be a blood loss consistently, very slightly over time. And here's two great examples down there, right? Hemorrhoids can cause anemia, chronic hemorrhagic anemia, because if you're bleeding consistently from these breakages of these blood vessels near around the anal sphincter, then this is where hemorrhoids are. Or sometimes you can have an ulcer, like in your stomach, where there can be blood vessels breaking and blood is actually pulling or leaking into the digestive system, and then you're losing that blood that way. Right? So there's many different ways that can happen. Those are just two, right? What about not getting enough red blood cells being produced? Well, we talked about this one, right? Iron deficiency, right? Anemia, which means you don't have enough iron in your blood, right? That could also be from the diet, not getting enough iron in your diet. or in your digestive system, you're not getting enough absorption of iron to your bloodstream. And as a result, your red blood cells, if you look at them under the microscope, will be really small in size and in color, right? Treatment is iron supplements, right? This is an interesting one. We call this pernicious anemia. And pernicious anemia is actually an autoimmune disorder or disease. The immune system is actually destroying the mucosa lining or the epithelial lining of the stomach. And the reason why we talk about this one is because the lining has glands that produce a chemical called intrinsic factor. Intrinsic factor is needed to absorb B12 in our body. And we said that B12 is important because it actually is involved in DNA production of a red blood cell. And so if you destroy the mucosa that produces intrinsic factor, you don't have intrinsic factor that absorbs B12, and if you eat food that has B12, it still goes straight through your body. It doesn't get absorbed. As a result, red blood cells grow in size, so they're much bigger than normal because they don't undergo division, because they don't have enough B12 to undergo DNA synthesis. Renal anemia can happen. This is because there's a lack of EPO, which means something's gone wrong with the kidneys. Kidneys are not secreting and producing enough EPO, and this is an issue there. Aplastic anemia discusses then the destruction of the red bone marrow, right? That can be from various different things, but sometimes viruses can do this, radiation, chemicals. We see this very commonly, for example, in cancer patients, because as they undergo treatment, they're trying to wipe away other cells. And one of the major side effects that is negative is that they strip away some of the red bone marrow cells that die. When they do that, they frequently can have aplastic anemia. And then sometimes we can have this issue where there's too many red blood cells that get destroyed. And we have these two under this umbrella, thalassemias and sickle cell anemia. Sickle cell anemia comes from the proteins that are misshapen, the globin proteins, in fact, that are misshapen. Because of that, oxygen can't bind to the hemoglobin molecule because it's misshapen, because the red blood cell is misshapen. And because of that, people that have sickle cell anemia have low oxygen levels. I'll show you this picture. We're running out of time here. So here is the part A. You can see then the amino acid sequence, where how many amino acids are put together and what order they're in, right? A hemoglobin molecule has 146 amino acids put together, right? And a normal one has 146, and we're listing just 1 through 7. There's an abbreviation for the amino acids, right? But I just want to look at part B. Go and look at part B and look at amino acid number 6. In part A, amino acid number 6 is glutamate. That's the abbreviation. And in amino acid number 6 in sickle cell anemia, it's been replaced by another amino acid that we call valine. Because that one amino acid gets switched, the red blood cell looks like part B instead of part A. That one amino acid. it misshapens just because of that one amino acid. Now, it's a genetic disorder, so sickle cell anemia is genetic. And so we can trace ethnicity, and African Americans have more of a risk of sickle cell anemia if your ancestors come from an area in Africa. Does that make sense to everyone? And that's why we see this genetic abnormality. Okay? So we're going to stop there, and the reason why we stop there is because we ran out of time here. But I do want you to cover the rest of it here, and we can. We didn't get to white blood cells, but I do want you to discuss the white blood cells, and in particular, there's information there, but when you start to take a look at those types of white blood cells, the percentages, right, is what you're going to concentrate on, and also looking at their composition and their function. Why do we have these white blood cells, and what are their function, right? Their structure and their function, right? What are they composed of, and what's their function? Right? Here's all your white blood cells on the right side. There's the percentages, but this talks about those type right there, all four of those. And I want you to discuss those. Okay? Let's go to